Good morning, everyone. Today, we embark on a journey to demystify the intricacy of past year exam questions for A-level students for October 2020. Examining the past offers us a compass to navigate the challenges ahead, empowering us with the insight and strategy to conquer the present. Let's dive into the treasure trove of past exams, unlocking the key to success and mastering the art of academic excellence. Which rows show the correct order of size of this cell structure? To determine the correct order of this cell structure from largest to smallest, let's look at the typical sizes of each structure. Width of the mitochondrion, typically about 0.5 to 1 micrometer. Width of ribosomes, normally is around 20 to 30 nanometer, which is 0.02 to 0.03 micrometer. Width of a cell surface membrane, approximately around 7 to 10 nanometers, which is 0.007 to 0.01 micrometer. Width of a chloroplast is typically about 4 to 6 micrometer. So now, chloroplast is between 4 to 6 micrometer. Mitochondrions is between 0.5 to 1 micrometer. Ribosomes is between 0.02 to 0.03 micrometer, and the cell surface membrane is about 0.007 to 0.01 micrometer. So the correct order from the largest to the smallest would be 4, 1, 2, 3. The diagram shows a stage micrometer scale view through an eyepiece containing a reticle. The small division of the stage micrometer scales are 0.1 millimeter. The stage micrometer scale is replaced by a slide of a plant cell. And what is the actual length of the nucleus in the plant cells? The answer will be 25 micrometer. Insulin is a hormone synthesized from two polypeptide chains inside cells of a pancreas. Which rows show the cell structures that are used in producing insulin and releasing it from the cells? Insulin productions and release involve rough endoplasmic reticulum, which is used for synthesizing insulin. Golgi body is used for processing and packaging insulin. And finally, lysosome is not used in insulin productions or release. The answer will be 1 and 2. So, lysosome, as we already explained, is not used in insulin. So the answer will be C. Number 4, the electron micrograph show a cell structure in a eukaryote cells. Which statements about these cell structures are correct? The answer will be the cell structure is made of protein molecule and the cell structure replicates during interphase of the cell cycle. 4 students were asked to match the functions with the appearance of some cell structures in an animal cells. These questions will be a very common questions and it's already appeared in May 2022. But in May 2022, the question is to list the functions. The functions listed is about mRNA, which is passes through the ribosomes, which produces mitotic spindle during cell division. And finally, it's also come into conclusion in terms of the packaging of hydrolytic enzyme that will remain in the cells. In the same thing, the same question, but except in this year, for October 2020, it focusing on the synthesis of polypeptide and synthesis of lipid. And the appearance were listed by letter, which is V, represent membrane which surround and enclose inner cavity. W is actually non-membrane bound spherical structures. X is a double membrane interspersed with pores, and Y is a non-membrane bound cylindrical structures, and Z is actually membrane bound sacs arranged as a flattened stack. And which student correctly matches the number functions with the appearance of the cell structure? The answer will be Number one is actually referring to W, which is non-membrane bound spherical structure. And number two is actually V, is a membrane which is surrounded and enclosed in a cavity. And number three is actually membrane bound sets arranged as a flattened stack. Number six, the DNA of typical prokaryote is naked and circular, which a statement describes how the DNA of eukaryotes differ from the DNA of typical prokaryotes. So, the DNA of eukaryote differ from that of prokaryote in two main ways. One, it has protein attached to it, which is forming chromatin. And number two, it is linear rather than circular. The answer will be D. Number seven, a student carried out the Benedict test on two different types of milks, X and Y. A sample of each type of milk was heated 200 Celsius in a water bath with Benedict solution, and the time taken for the first appearance of the color change was recorded. Which row shows the biological molecule the student detected in each sample of milk and the sample of milk with the highest concentrations of this biological molecule? The answer will be C, reducing sugar, and the time for first appearance of the color change with the Benedict solution in second will be 13. Number 8, the diagram shows the structure of monomer. Which molecule contains this monomer? The answer will be aminopeptide, glycogen, and sucrose. Number 9, which molecules are formed by condensation which involve a carboxylic group? The answer will be these molecules are formed through condensation reaction which is involved carboxylic group. Will be amylopectin, collagen, and triglycerides. Which row correctly matches the function with the structure features of cellulose, collagen, glycogen, or triglyceride? The answer will be molecule referring to cellulose. Function is for support, and fibrous will be correct, and molecule bound together by hydrogen is also correct, and branch change is not correct. But for the triglycerides, function is as energy source. For the fibrous, is not correct, and for the molecule bound together by the hydrogen bond also not correct, and the branch change also not correct. Number 11, the diagram shows the amino acids in a polypeptide. An enzyme catalyzes the hydrolysis of any peptide bond between the non-polar amino acids and any polar amino acid. How many small peptides and single amino acid will be formed by the actions of this enzyme? The answer will be G. Small peptides will be 13 and the single amino acids will be 4. Which properties of water reduce temperature changes inside cells? The answer will be only 3, which is specific heat capacity, because the high specific heat capacity of water had reduced temperature changes inside cells. Which enzyme is extracellular? The answer will be A. Amylase in saliva is extracellular enzyme because its function outside of the cells, specifically in the mouth, to catalyze the breakdown of starch into simpler sugar. This contrasts with intracellular enzyme like ATP synthetase, DNA polymerase, and RNA polymerase, which function inside the cells. The graph shows how the concentration of component of an enzyme catalyzed reaction changes with time. Which line represents enzymes anti-active site? The answer will be D. Which effect can non-competitive inhibitors have an enzyme control reactions? The answer will be reduce the concentration of the product and reduce the rate of metabolic pathway. Non-competitive inhibitor reduce the concentration of the product and reduce the rate of metabolic pathway by binding to an enzyme at a site other than the active site, altering the enzyme functions without affecting the KN value. Which processes can allow transport into or out of the cells? The answer will be active transport, facilitated diffusion, and osmosis. These are the processes that can allow transport into or out of the cells. Plant cells were put into one of three different concentrations of sugar solution, 10%, 5%, and 2.5%. And the cells were left for 50 minutes and then observed using a light microscope. Which statements are correct? The three statements are correct. Cells Y have a lower water potential than the sugar solution it was put into. Cell Z was put into the 10% sugar solution, and cell Z had a less negative water potential than the sugar solution it was put into. During telophase of mitosis, scientists stain the chromosomes of the diploid animal cells with a fluorescence dye to observe the telomeres. These cells have 22 chromosomes. How many telomeres will the scientists observe in one of the nuclei? Each chromosome has 2 telomeres, one at each end. In a diploid cells with 22 chromosomes, there will be a 22 chromosome multiplied by 2 telomeres per chromosome multiplied by 2 nuclei. And the answer will be 88 telomere observed in one of the nuclei. Methotrexate is a drug used to treat cancer. It can act as an enzyme inhibitor, preventing the synthesis of nucleotides containing thymine. Cells treated with methotrexate are not able to complete the cell cycle. A cell entering telophase is treated with the methotrexate. Which stage of the cell cycle will be affected by the actions of methotrexate? The answer is interphase. Methotrexate inhibits the synthesis of nucleotides containing thymine, which are necessary for DNA replication. DNA replication occurs during the S phase of interphase. Therefore, interphase will be affected by the actions of methotrexate. Which stage of mitosis is correctly described? The answer will be C, which is in prophase, chromatin move to opposite poles. Let's look into the explanation why the answer is C. Anaphase, during this phase, chromatin which are now separated sister chromatin move to opposite poles of the cells. Metaphase is a phase whereby centrosomes replicated during interphase, not specifically during metaphase. And in telophase, chromosomes uncoil and decondense to form chromatin as the nuclear envelope will form around them. Therefore, the answer C is accurately described an event that occurs during anaphase or mitosis. Question number 21 is a common question which is also already exists in March 2022. But in that question for March 2022, the question is asking about adenine. It's not cytosine. So in this year, for the October 2020, the question is asking about cytosine. So which statement about these nucleotides are correct? The carbohydrate could be ribose. The organic base contain nitrogen, and base pairing occurs with guanine. And cytosine is a purine. The answer will be 1, 2, and 3. A section of DNA contains 73 base pair. Which row represent the correct number of bases? Let's see the next page for further explanation. So to determine the number of each type of base on each DNA strand, we start by analyzing the given information. So the information given here is strand 1, adenine, taken the value of 29, cytosines, number of bases on each strand is V, we do not know. Guanine is 14, and thymine is W. For the strand 2, we have adenine 21, cytosine X, guanine Y, and thymine Z. So we know that the total number of base pair in the DNA section given in the question is 73. Therefore, the sum of bases on both strands should add up to 73. Let's assign variables to V, W, X, Y, and Z based on the information provided. From strand 1, we can see that V plus W is equivalent to 73 minus 21 plus 14. Then you get the value of 30. That is referring to strand 1. From strand 2, X plus Y plus Z is also 73, and minus by 21, you get a value of 52. So now using the answer choices, we need to find the value that satisfies this equation. Let's evaluate each option. Option A, given V is equal to 9, W is equal to 21, X is equal to 14, and Y equals to 9, and Z is equal to 29. Let's recheck. Strand 1, V plus W is equal to 9 plus 21. You get the value of 30. Strand 2, X plus Y plus Z is equal to 14 plus 9 plus 29. You get the value of 52. So both conditions are satisfied with option A. Therefore, option A correctly represents the number of bases on each strand. So strand 1, A is equivalent to 29, C is equivalent to 9, and G is equivalent to 14, and D is equivalent to 21. And for the strand 2, you have the value of A is equivalent to 21, C is equivalent to 14, and G is equivalent to 9, and T is equivalent to 29. So the correct answer is an options given in the answer, which is V is equal to 9, W is equal to 21, X equal to 14, Y is equal to 9, and Z is equal to 29. The photograph shows a section of a stem, which label part is the xylem. So in this diagram, C is actually represent the xylem. Some fungi cause wicking in plants by growing within the xylem vessels element, which process will be directly affected by this fungi. So the process directly affected by the fungi growing within the xylem vessels element is cohesion between water molecules. This is because the fungi block the xylem vessels, disrupting the continuous flow of water that relies on the cohesion between water molecules. And this blockage leads to wilting in plant, which features have a role in the transport of water in xylem vessels element. So the answer will be capillary reaction, excision, and hydrogen bonding, 1, 2, and 3. Capillary action helps water move outwards in narrow xylem vessels. Adhesion allows water molecule to stick to the wall of the xylem vessels, and hydrogen bonding contribute to the cohesion water between molecules, allowing them to move together in a continuous stream. The loading of sucrose into companion cells involve a number of processes. Which processes is active? The answer will be the movement of hydrogen ion out of the cytoplasm of companion cells. This is because active transport farm hydrogen ions out of the companion cells within a proton gradient. And this gradient is then used to co-transport sucrose into the companion cells through a symporter, which is a passive process driven by the proton gradient. 27. The statement describes event that occurs during the movement of sucrose. So the correct sequence of events that describe what happening in a plant at the source during the movement of the sucrose will be: sucrose is loaded into the Follium vessels. Sucrose is loaded into the Follium vessels at the source. Example, leaf sucrose is actively transported into the Follium. Water potential decrease in the Follium. The addition of sucrose lowers the water potential in the folium because sucrose is a solute. And the water moves by osmosis into the folium because the water potential is now lower in the folium compared to the surrounding cells. Water moves into the folium by osmosis. And number 4 will be, and number 2 will be hydrostatic pressure is increased. The influx of water increases the turbo pressure, hydrostatic pressure in the folium, which helps to drive the flow of the folium set. The answer will be 3, 7, 5, and 2. 28, which are present in the wall of capillaries. The correct option for the structure present in the wall of capillary is endothelium. Capillaries are composed of a single layer of endothelial cells and lack smooth muscle, elastic tissue, and collagen fiber. And this thin structure facilitates the exchange of gases, nutrients, and waste products between the blood and surrounding tissue. The photomicrograph shows 3 white blood cells labeled as X, Y, and Z, which will correctly identify these cells. This common question is already exists in May 2023. The answer for this question would be monocyte is a cell's X, cells Y represent neutrophil, and cell Z represent lymphocytes. Number 30 question is a common question, exists in May 2023. And which sequence letters correctly identify the order of event during the cardiac cycle? The correct sequence of event during the cardiac cycle is as follows. First is a X, which is wave of excitation spread from the sinoatrial nodes, and then followed by T, atrial walls contract, and then followed by V, wave of excitation enter the atrioventricular nodes, and then followed by U, impulse is delayed by a fraction of a second, and then W, wave of excitation passes down the Purkinje fiber, and then Y is ventricle contract. So the correct sequence will be X, T, V, U, W, and Y. The graph shows the effect of 3 different partial pressures of carbon dioxide on the oxygen association curve for human hemoglobin. What effect does decreasing the partial pressures of carbon dioxide have on hemoglobin? It is more efficient at taking up oxygen and less efficient at releasing oxygen. Number 32, what is correct about the transport of carbon dioxide by the blood? The correct options for the transport of carbon dioxide by the blood would be 1 and 2, which is the enzyme carbonic hydroxy catalyze the formation of carbonic acid in red blood cells. And number 2 will be carbon dioxide diffuses from active cells to red blood cells and react with water. And this first and 2 is the correct options. But the third statement, carbonic acid is associated from hydrogen ion that combine hemoglobin to form carbaminohemoglobin is incorrect because carbaminohemoglobin is formed when carbon dioxide directly binds to hemoglobin, not formed from the dissociation of carbonic acid. The dissociation of carbonic acid produces hydrogen ion and bicarbonate ions with the hydrogen ion binding to hemoglobin, but this process does not form carbaminohemoglobin. 33, which statement about all bronchioles are correct? The correct is only they have slated cells, and number 3, they have muscle tissue. And bronchioles contain slated cells that have immune mucus and trap particles out of the lung. They also have smooth muscle tissue that allow them to contract and dilate, regulating airflow to the alveoli. However, not all bronchioles have goblet cells. Goblet cells are more commonly found in the large airways such as trachea and bronchi. The graph shows the volume of air breathed out quickly and with force following a deep breath in for 3 different peoples X, Y, Z. What is a possible explanation for differences in the volume of air breathed out by these people shows? The answer will be C. 35. The diagram shows some of the pathogen that cause diseases in humans and some of the way they are transmitted. This is a very common question, and also the same question exists in May 2023. What is the correct pathogen and methods of transmission for the disease TB? There will be 2 and X. 36. The proportion of the local population who have malaria in that area R is higher than the proportion in area S. What factors causes these differences? The differences is area R has a more humid climate than the area S. A more humid climate can lead to a higher incidence of malaria because it provides a favorable environment for mosquito, which are the vectors for malaria. Species X is a single-cell eukaryote. Species X has been genetically modified to produce penicillin, which does not harm the cell wall of species X. What may be concluded from this information? The answer is only one. The cell wall of species X are chemically different from those of bacteria, as penicillin target bacteria cells wall made of peptidoglycan and does not harm species X. 38. When exposed to an antigen for a second time, memory cells stimulate a secondary immune response, which correctly show the secondary immune response. So the answer will be divides into plasma cells and memory cells. The answer will be C. Monoclonal antibodies are produced for use in diagnosis or treatments of disease. To obtain the antibodies for an antigen, a mouse is injected with an antigen. Some of the events in the productions of the monoclonal antibodies are listed. To determine the correct sequence of the first 4 events in the productions of monoclonal antibody, we need to understand the logical orders of this step. First, followed by B lymphocyte that recognize the antigen, multiply and become plasma cells. And then followed by the step number 5, plasma cells are removed from the mouse skin. And then step number 1, plasma cells are fused with cancer cells to form a hybridoma. And finally, step 2, which is hybridomas that separates the required antibodies are identified and cloned. Number 14, which role describes passive immunity? Passive immunity is when antibodies are given to a person rather than produced by own immune system. And it does not involve an immune response, does not produce memory cells, and does not offer permanent protection. Therefore, the correct answer will be D. That's all for today's presentation. And as a summarization from what we have learned today, by leveraging past examination paper, you can enhance your understanding, improve your exam technique, and ultimately achieve better results. So, last but not least, thank you for your attention and best of luck in your studies and upcoming examination. Please don't forget to subscribe to Jom Study Now YouTube channel.